Hello, it's Rodina Nagar, and today we're going to be solving October-November 2021, Paper 1, Variant 3. Question number 1. A student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. Which method could the student use to calculate the magnification of these cells? Here we have the actual size in micrometers, and the equation for magnification is I am. And we're trying to work out the magnification. So what we do is we divide the image size by the actual size, but we have to convert the image size into micrometers. Therefore, the answer that actually suits this is C, because here it mentions measure the length of the scale bar in millimeters, convert to micrometers by multiplying by 1000, and divide by 2. Divide by 2 is the actual size in micrometers. And there you have the magnification. Question 2. Which statement about light microscopy are always correct? 1. The greater the resolution of a light microscope, the greater the detail that can be seen. This is obviously correct because the resolution is the ability to distinguish between two separate points. And the shorter the wavelength of the light, the greater the resolution. Number 2, the greater the magnification of a light microscope, the greater the detail that can be seen. This is incorrect because it's always up to the limit of resolution. Coming to point number 3, and it's increasing the magnification of a light microscope up to its limit of resolution allows more detail to be seen. This is totally correct. Point 4, the shorter the wavelength of light used in a light microscope, the greater the detail that can be seen. We just mentioned about this, and this is correct because the shorter the wavelength, the greater the resolution. Therefore, the answer is going to be B. Question number 3. What is the length of a typical prokaryote, such as this one? A length of a typical eukaryote is always from 0.1 to 5 micrometers. The only answer within this range is going to be C. Question number 4. Which cell structures are present in Plasmodium? Plasmodium is the eukaryote responsible for malaria, and here are the 4 different species of Plasmodium, okay? And since we just mentioned that it's a eukaryote, then it definitely does have ribosomes and mitochondria and Golgi body. So the answer is A. Question number 5. Which cell structures may contain nucleic acids? And now these are examples of nucleic acids: DNA, mRNA, and tRNA. In the cytoplasm, we know we have mRNA and tRNA, so this is correct. In the chloroplasts, we know we have circular DNA, so it does exist, and this is correct. 3. In the ribosomes, we have mRNA that occurs during translation. This is correct. And point number 4, Golgi body is incorrect because Golgi body packages and modifies protein and it has nothing to do with nucleic acid. So the answer is going to be A. Question number 6. Which row shows correct features of ATP? Now right here we have the structure of ATP, and it's 3 phosphates right here and a ribose sugar and adenine. Here we have carbohydrate in ATP. The first one we have is deoxyribose. This is totally incorrect. This is the carbohydrate actually found in DNA nucleotides, not ATP. For B, we have pentose. This is correct because ATP has a 5-ringed glucose. For C, it shows hexose. Hexose is definitely incorrect because you just mentioned it's a pentose sugar. And D, we have ribose. Ribose is correct because it's an example of a pentose sugar. Now, the site of production of ATP. Mitochondria and chloroplasts both are correct. Chloroplasts only are incorrect, and this is incorrect. Therefore, the answer is going to be B. Now, gladly we won't be doing this question since it has been discontinued because it just did not make any sense. Question 8. Which polysaccharide is correctly described? A. Amylopectin monomer and it's beta glucose. This is incorrect. Actually, it's alpha glucose. For B, polysaccharide amylose, and it's correct. It has alpha glucose monomers, and its biological role is storage molecule in plants. This is correct. Let's see the rest. Cellulose. It assumes here it's alpha glucose. This is incorrect. It's actually beta glucose. And it has a structural role in plants. This is correct, actually, this statement. Polysaccharide glycogen. And it does not have beta glucose. And it is a structural molecule in animals. This is correct. So the correct answer is B. Question number 9. Which formula represents a saturated fatty acid? Here I've given you the formula of a saturated fatty acid. It's CnH2nO2. The only answer that satisfies this criteria is A, because if you multiply 18 by 2 is going to give you exactly 36, which is 2n. So the answer is definitely going to be A. Question 10. Which molecules always contain at least 4 double bonds? So, to start with, it's definitely going to be a protein because proteins contain carboxylic acid groups, and these definitely do have double bonds. So let's check what we have here. We have triglyceride. This is incorrect. Collagen, yes, this is correct. Hemoglobin is a protein, so this is correct. Obviously, proteins are made up of amino acids. For B, collagen, this is correct. Triglyceride, no, this is incorrect. For C, hemoglobin is a protein and collagen is a protein, so this might be correct. Triglyceride, this is incorrect. Hemoglobin, this is correct. So the answer is going to be C. Question 11. Which row about the structure of proteins is correct? To start with, the primary structure. The number of amino acids present in a protein. It's not actually the number. It depends really on the order of amino acids or the type of amino acids. The order of amino acids present in a protein encoded by DNA. This is correct. For C, the result of translation of an mRNA molecule by ribosome into a chain of amino acids. This is correct. D, the sequence of amino acids in a protein encoded by an mRNA molecule. And this is also correct. For the secondary structure, the left-handed spiral formed by the primary structure. Here by the left-handed spiral, it basically means an alpha helix. So this is correct because secondary structure is hydrogen bonding, forming either an alpha helix or a B-pleated sheet. B, the coiling of a chain of amino acids to form a B-pleated sheet. This is correct. C, occurs because of attraction between hydrogen and oxygen atoms in side chains. Obviously, this is incorrect because secondary structure occurs because of the attraction between hydrogen atoms in the amine group of the amino acid itself, of the peptide bond, okay? And the oxygen atoms in the carboxylic acid group also in the peptide bond. So mentioning the side chain is definitely incorrect. D, formed by hydrogen bonding between amino acids forming the primary structure. This is definitely correct. Tertiary structure, the result of cross-bonding between specific amino acids in the primary structure. Obviously, this is correct. Tertiary structure actually includes bonds formed between the R groups. The first type of bond being disulfide and hydrophobic, hydrogen, and ionic. And same goes for also quaternary structure. B, shape formed by folding of a polypeptide and held together by hydrogen bonds. Obviously, hydrogen bonds is incorrect because here we've mentioned 4 different bonds. The result of ionic and hydrogen bonds, disulfide bridges, and hydrophobic interactions between amino acids. This is definitely correct. Formed as a result of interaction of side chains of amino acids of primary structure. Yes, that is also correct. Finally, the answer is D. 12. Which statement about collagen is correct? A, a collagen fiber is made of 3 parallel helices with hydrogen bonds holding them in place. First of all, the totally wrong information is fiber, because if a 3 helices, basically the 3 polypeptide chains holding them in place, it forms a collagen molecule. not a fiber. This is totally incorrect. B. It is an insoluble fibrous protein with a quaternary structure. Let's start with insoluble. Insoluble is definitely correct because it's a structural protein and not metabolically active. Okay? And it's fibrous. Quaternary structure is correct because it's formed by 3 polypeptide chain held together by hydrogen bonds. C. 1/3 of the amino acids making up collagen are valine. Valine obviously is incorrect, it's actually glycine. Every third amino acid is glycine, and there are proline as well. So this is incorrect. The collagen fibers are formed from several collagen molecules held together by ionic bonds. Ionic is incorrect because it's either hydrogen and some covalent bonding. So the final answer is going to be B. 13. Which properties of water are the result of hydrogen bonding between water molecules? Okay, the first point, solvent action is actually correct and it is a result of hydrogen bonding. However, it's not between water molecules, it's between solute molecules. Therefore, in this case, it's going to be incorrect because it's not addressing the question. Point number 2, specific heat capacity. Specific heat capacity is the amount of energy required to increase the temperature of water by 1 degree. And hydrogen bonding makes water require incredibly so much energy in order to break the strong bonding. So this is correct. Latent heat of vaporization is the energy required to vaporize that water, and as a result of strong hydrogen bonding, really high energy is required. So this is correct. Therefore, the answer is going to be C. Question 14. Which descriptions about enzymes are correct? Number 1, catalyze the breakdown of large molecules into smaller molecules. Okay, this is correct, however, not for all enzymes, because some enzymes carry out anabolic functions and catabolic functions. Catabolic is breaking down molecules into smaller ones, and anabolic is building molecules from smaller ones. So this is incorrect. 2, only function inside cells. This is also incorrect because we have intracellular enzymes and extracellular enzymes. 3, form temporary bonds with the substrate. This is correct because once the substrate enters the active site of the enzyme, bonds are formed between them in order to break the substance down. So this is correct. 4, have a tertiary structure. Definitely all enzymes do have a tertiary structure because this structure and this type of bonding is what actually forms the active site of the enzyme and what determines the shape of the active site. Therefore, D is going to be correct. 3 and 4. Question number 15. The cells in the roots of beetroot plants contains a red pigment. When the pieces of root tissue are soaked in cold water, some of the red pigment leaks out of the cells into the water. An experiment was carried out to investigate the effect of temperature on the loss of red pigment. So here, the temperature on the loss of red pigment from the root hair cells. It was found that the higher temperature of the water, the higher the rate of loss of red pigment from the root cells. Which statement could explain this trend? So let's first summarize this experiment really quickly and explain its factors. Here it's experimenting diffusion. As we all know, the rate of diffusion increases with increasing temperature. Why? Because particles now have more kinetic energy and they move faster. Okay, so let's start with the first point. Enzymes in the cells denature as the temperature increases, so the pigment can no longer be used for reactions inside the cell and diffuses out. Obviously, this is totally incorrect. Okay, the part about enzymes denaturing is correct, but the rest of the question just does not make any sense. Part 2, the temperature increases, the tertiary structure of protein molecules in cell surface membranes changes. Yes, this is correct because protein molecules denature because high temperature alters the hydrogen bonding in the tertiary structure. Okay, increasing the permeability of the membrane. This is correct. 3. Phospholipid molecules gain kinetic energy, what we just mentioned, as the temperature rises, increasing the fluidity of the phospholipid bilayer and allowing pigment molecules to diffuse out more easily. This is correct. Increased temperature increase the fluidity of the cell surface membrane, allowing more substances to leak out. So the answer is going to be B, which is 2 and 3. Question number 16. The diagram shows a partially plasmolysed plant cell. Plasmolysed, why is it plasmolysed? Is because the exterior fluid has a lower water potential than the inside fluid, which has a higher water potential. So water moves out by osmosis down water potential gradient. Here it's asking what is found at Z. Okay, once the plant is plasmolysed, the cell surface membrane shrinks inside the plant cell while the cell wall stays in place because it has a supporting material such as cellulose. If you don't know, the cell wall is actually freely permeable, meaning any substance could pass through, any substance could possibly pass through. So here we have solution Y. Once it's plasmolysed, solution Y could easily go inside the plant cell. So C is going to be answer. Question number 17. A single-celled organism lives in fresh water. Fresh water, the first point is that it has a really high water potential. Okay, water that enters the cytoplasm of the cell by osmosis is collected into a structure called the contractile vacuole. To remove the water, the contractile vacuole fuses with the cell membrane. A student counted the number of times that the contractile vacuole filled and emptied when the cell was placed in solutions with different water potentials. The results are shown in the table. Here we have the water potential of external solution. It's a common misconception. So here, we can see that the water potential gets more negative. Water potential getting more negative basically means water potential is decreasing. Here in the second column, you can see the rate of contractile vacuole emptying. And as we see, when the water potential decreases, the rate of contractile vacuole emptying decreases too. This gives us a sign that the single-cell organism also has a really low water potential, because if the contractile vacuole is emptying way too much, this means that so much water is entering inside the cell. But here, if it's the rate of contractile vacuole emptying only a little bit, it means just quite less water is entering the cell. Here we have 0, so the rate of contractile vacuole emptying is 0. It means it's not emptying anymore. Therefore, water stopped entering the cell. So most likely the water potential of the single-celled organism is going to be negative 500. Okay, let's now read the rest of the question. Which statement explains the pattern observed as the water potential of the external solution decreases? Okay, so the only answer that suits our explanation is going to be D. The reason for this is it mentions the water potential gradient between the cell and the solution decreased, causing water to move into cells less rapidly. We just mentioned this above, and the contractile vacuole to empty less frequently. So the answer is D. Question number 18. Which row shows the correct number of each component of a single chromatid during anaphase? Okay, so during anaphase, the spindle fiber pulls the two sister chromatids apart to opposite poles, and that's what they both look like. So it only has, in this case, it only has one centromere. Each chromatid only has one centromere, so this is correct. And here, polynucleotide strand. We know that each chromatid has actually two polynucleotide strands or a single DNA molecule. I'm going to write it down. Two polynucleotide strands or a single DNA molecule. Because we know a single DNA molecule is made of a double helix. So 2 is correct. For telomeres, we know that each end of a chromatid has a telomere. So each chromatid has 2 telomeres. I'm going to write this down. Each chromatid has 2 telomeres. So I'll give you an example. If now we have a single chromosome which has 2 chromatids, is going to actually have 4 telomeres. But in this case, because we're only talking about 1 chromatid, it's going to be 2 telomeres. So the answer is A. Processes are listed: cytokinesis, differentiation, DNA replication, and mitosis. How many of the listed processes occur during tissue repair by stem cells? First of all, for stem cells to do tissue repair, they have to divide. Divide how? By undergoing cell cycle. Now, the cell cycle includes cytokinesis, and it's the stage that comes after mitosis. Differentiation is the ability to divide into any type of cell. This is correct, and that's how it actually repairs the tissue. 3. DNA replication. This is correct because the full cell cycle has an S phase. The S phase is where semi-conservative DNA replication occurs. And the 4th point being mitosis. Mitosis is also a part of the cell cycle where prophase, metaphase, anaphase, and telophase take place. So all 4 are correct. So the answer is going to be D. 20. The photomicrographs show cells in various stages of the cell cycle. Which stage of mitosis is not shown? Let's analyze this. Here we can see condensed chromatids, which are clearly visible to us. This stage is actually prophase. If you see condensed chromatids like this, it's definitely going to be prophase. The cell beside it has chromatids clearly shown at opposite ends of the cell, so this might be either late anaphase or early telophase. Here we could clearly see telophase taking place. Actually, late telophase leading to cytokinesis. And lastly, this cell shows anaphase. So if we see here, clearly the missing stage is going to be metaphase. And quickly, metaphase is the lining of chromosomes across the metaphase equator for spindle fibers to attach to the centromeres of the chromosomes to pull them apart. 21. Which letter in the key describes uracil? So, let's just make things clear. Purines are AJ, adenine and guanine, which is a double ringed structure. So, double ringed. However, pyrimidines are cut, cytosine, uracil, and thymine, which is a single ring. That's how you should memorize it. So, coming back to the question, uracil, it's definitely not a purine. Has a single ring structure, yes. So the answer is going to be C. 22. When a gene for protease is activated, which nucleic acid will be formed? Okay, so we know that if the gene protease is activated, it's a protein, and therefore transcription is going to take place. And transcription happens when mRNA is formed to go to the ribosome to get translated, to translate that gene into a polypeptide. So the answer is definitely going to be B. Question 23. The diagram shows a vascular bundle from the stem of a plant. Which row describes the functions of the labeled cells? First of all, let's start with the table. Transport sucrose. We all know that Phloem sieve tube elements is what transports sucrose. Transports mineral ions. We all know that xylem vessel elements is what transports water and mineral ions. Third one is stores starch. Here, I've written that parenchyma cells is what stores starch. So let's see what labels we have here. For label 1, it's definitely going to be the xylem vessel elements. Number 1, why would you ask? Because it has a lignified cell wall, and definitely if it has a lignified cell wall, then it's a xylem. Second of all, it's actually larger than the rest of the cells in that same region, so it clearly shows that it's a xylem. So 1 here is correct. Second of all, for label 2, it's definitely going to be a phloem sieve tube element, because you can see it's way smaller than the lignified xylem vessel elements. So it's going to be 2. For store starch, parenchyma cells are always on the outside and they look way larger than the rest. So 3 is definitely going to be parenchyma cell. So the answer is going to be B. Question number 24. Some of the features present in the transport tissues of plants are listed. Which features are present in sieve tube elements? The first point is lignified walls. Because sieve tube elements are living cells, they are unlignified. What's lignified is xylem vessel elements. So this is incorrect. 2, cytoplasm. Yes, they do have cytoplasm, however, a peripheral cytoplasm only at the edges of the cell. So it's not full of cytoplasm, but this is correct. 3, mitochondria. Yes, Phloem sieve tube elements definitely has some mitochondria. This is true. Point number 4. No, they do not have chloroplasts. This is incorrect. 5, plasmodesmata. Definitely they do have plasmodesmata. So sucrose can move from companion cells into the phloem sieve tube elements through this plasmodesmata. So the correct answer is 2, 3, and 5. So the answer is C. Question number 25. Which statements explain why transpiration is an inevitable consequence of gas exchange in plants? We all know that transpiration happens, and transpiration is loss of water vapor through the stomata. So when the stomata opens, for water vapor to go out of the stomata, gas exchange takes place. Therefore, the only correct answer here is number 3, because hydrolysis reaction has nothing to do with the question, and same as ATP production, the only correct answer must be D. In an investigation, a leafy shoot was attached to the potometer and exposed to a variety of conditions. The time taken for the meniscus to move 50 millimeters along the capillary tubing was recorded for each set of conditions. Which row shows the slowest and fastest rates of water uptake for this investigation? In the table here, we could see a column saying time taken to move 50 millimeters per second. So obviously the slowest rate is going to be the one that took the most time to move. And in this case, it's going to be 166. And the fastest rate is definitely going to be the one that took the least time to move. In this case, it's 40. So what do we do to calculate the slowest rate? Is that we divide 50 by 166. This would give us 0.3. So 0.3 is the correct answer. And then for the fastest rate, we would divide 50 by 40. This would give us 1.3. So 1.3 is correct. The only correct answer should be A. 27. Proton pumps and co-transporters are used by plants when loading sucrose into a phloem sieve tube elements at the source. Which row is correct? So first, I'm going to explain the mechanism of the proton pump in the companion cell. First of all, here we have a companion cell. At the cell membrane, there are proton pumps. These proton pumps, what they do is they pump out H+ ions out of the companion cell into the companion cell wall. We're good with this point? So, proton pump, let's see which answer should be correct. Pumps protons into a companion cell, raising its pH. Actually, pumping protons into a companion cell does not raise its pH, it actually lowers the pH, because the higher concentration of H+ ions, the lower the pH. So this is incorrect. Pumps protons into a mesophyll cell, raising its pH. Mesophyll cells have nothing to do with the mechanism right here. Here we're talking about companion cells. So this is incorrect. C, pumps protons out of a companion cell into its cell wall. This is definitely correct, and that's what we said right here. D, pumps protons out of a mesophyll cell. Mesophyll is incorrect. Let's see the other column. Co-transporter. Now what the co-transporter does is there are sucrose at the outside of that companion cell. What happens is both H+ ions and sucrose goes back inside the companion cell through a co-transporter protein by facilitated diffusion. Sucrose goes against concentration gradient and H+ ions goes down concentration gradient. So let's start with the first one. The high concentration of protons in the companion cell allows the transporter to be used for moving sucrose out of the cell. Sucrose does not move out of the cell. This is incorrect. The high concentration of protons in a mesophyll cell drives mesophyll is also incorrect. The high concentration of protons outside a companion cell drives the transport of sucrose into the cell. This is definitely correct. The answer is C. The statements list some of the events in the cardiac cycle. They are not in correct order. Which statements describe the 6 of these events to occur in the cardiac cycle? To start with, we start with 4, the sinoatrial node contracts. So what the sinoatrial node does, it's in the right atrium and initiates the excitation wave. So this is the first one. Number 2, a wave of excitation sweeps across the atria. The wave travels across the atria. This is correct. Number 3, the atria contract. Once the wave travels across the atria, it contracts. Then what happens is that the atrioventricular node delays the impulse for a fraction of a second. It's actually 0.1 seconds. So this is the fourth. The impulse travels through Purkinje tissue. Purkinje tissue is found at the base of the ventricles. So this is the fifth. Then the sixth is going to be the wave of excitation sweeps upwards from the base of the ventricles, because once it reaches the Purkinje tissue, it sweeps upwards. So this is definitely going to be the sixth. So the sixth is going to be D. 9. Which tissue types are present in walls of all blood vessels? So to make things clear, the blood vessels that we have are arteries, capillaries, and veins. The tissue types that are present in walls of all blood vessels is only going to actually be an endothelial. Why? Because collagen, elastic fibers, and smooth muscle are only present in arteries and veins. They are not present in capillaries. However, in arteries, they are present in higher proportions than veins. Veins have lower proportion of these. So the only correct answer is going to be D. The photomicrograph shows 3 white blood cells labeled X, Y, and Z. Which row correctly identifies these cells? To start with, we have here cell Y. It looks like almost a bean-shaped nucleus. This bean-shaped nucleus is definitely going always to be a monocyte. So this is correct. For cell X, here we can see an almost granular nucleus. This granular nucleus is definitely always going to be a neutrophil. This is correct. And for Z, we can see that the nucleus occupies almost the whole cell, and this must be a lymphocyte if it occupies the full cell. 31. When active tissues have high carbon dioxide concentration, oxyhemoglobin needs to release oxygen to the tissues. How is the carbon dioxide transported away by the blood from these tissues? Number 1, as carboxyhemoglobin. This is incorrect. Because carboxyhemoglobin means that carbon monoxide has binded to the hemoglobin, not carbon dioxide, so this is definitely incorrect. 2 as carbaminohemoglobin. This is correct. Carbaminohemoglobin happens when carbon dioxide binds with the globin of the hemoglobin in the terminal amine groups, forming carbaminohemoglobin. This is correct. Point number 3 as hydrogen carbonate ions. So, in the red blood cell, carbon dioxide combines with water to form carbonic acid. This reaction is facilitated by an enzyme called carbonic anhydrase enzyme. Once carbonic acid is formed, it dissociates into hydrogen carbonate ions and H+ ions, and the hydrogen carbonate ions moves out of the red blood cell into the plasma. Therefore, this is definitely correct. The answer is going to be C. Question number 32. Which effect does increasing carbon dioxide concentration have on hemoglobin? As we mentioned earlier in the previous question, when carbon dioxide concentration increases, this means that carbonic acid concentration increases. Therefore, hydrogen carbonate concentration increases and H+ ion concentration increases. When this happens, H+ ions binds to hemoglobin, forming hemoglobinic acid. Hemoglobinic acid has a much lower affinity for oxygen. Therefore, hemoglobinic acid releases oxygen way easier and combines with oxygen more harder. Now let's read the choices. Hemoglobin is less efficient at taking up oxygen. This is correct. And less efficient at releasing oxygen. It's not less efficient, as I said, it has a lower affinity, so it's actually more efficient at releasing oxygen. B. Hemoglobin is less efficient at taking up oxygen and more efficient at releasing oxygen. And this is definitely correct. 33. The diagram shows a magnified section of part of lungs containing specialized tissues. Which row is correct for the structures labeled 1 to 6? So let's break this down. I know it could be a bit confusing. To start with, this structure you see here is definitely a red blood cell. Around it is the capillary wall. And this is the alveoli. Inside the red blood cell is going to be the carbonic anhydrase enzyme. Let's see the choices we have here. Choice number 1, carbonic anhydrase, it's assuming that is going to be the squamous epithelium. Here's the squamous epithelium. And this is the lining of the alveoli. So this is definitely incorrect. 2, it's assuming that the capillary wall is the carbonic anhydrase, and this is incorrect. For C, here we could see 3. 3 is inside the red blood cell. Definitely carbonic anhydrase is found inside the red blood cell, so this is going to be correct. And 4 is the air space inside the alveoli. It definitely does not contain carbonic anhydrase, so this is incorrect. Let's continue with C because it seems to be the only correct answer. 5 is assuming that it has a high proportion of hydrogen carbonate ions. Once carbonic acid dissociates into hydrogen carbonate and H+ ions, the hydrogen carbonate leaves the red blood cell into the plasma, and hydrogen carbonate is found in high proportion in the plasma. So that 5 is definitely correct. Lysosomes, it's assuming that it's 6. Lysosomes are single-celled organelles found inside the macrophage, which digest any pathogen engulfed by the macrophage. Here is actually a macrophage. This is definitely correct. The answer is going to be C. 54. The table shows some parts of an animal's lungs that contain different cell types. Which parts of the animal's lungs clean inhaled air and which carry out gas exchange? So let's start with the first part. Clean inhaled air is definitely going to be a structure which contains both goblet cells and cilia. Goblet cells release mucus, which traps dust particles, and cilia sweeps that mucus to the back of the throat so it can be swallowed. This is the mechanism lungs use to clean the inhaled air. And gas exchange is definitely carried out in the alveolus, where gases move against down concentration gradient. Therefore, we know that bronchioles have a small proportion of goblet cells. Bronchus has both goblet cells and cilia, and trachea both also has goblet cells and trachea, so this is correct for clean inhaled air. Gas exchange is going to be alveoli because this is the gas exchanging site in animals. So D is definitely going to be the correct choice. 35. What helps to maintain a steep oxygen concentration gradient between the air in the alveolus and in the blood? Breathing in brings in a supply of air, a relatively high concentration of oxygen to the alveolus. This basically means that it's going to be a concentration gradient difference, so oxygen moves into the squamous epithelium cells of the alveoli, down concentration gradient into the blood capillaries. So this is definitely correct. Point 2. Blood flow brings blood with a relatively low concentration of oxygen to the alveolus. It literally implies the same points as before, that the capillaries now have a lower concentration of oxygen. Therefore, there's also going to be a concentration gradient, and oxygen goes from the air spaces to the blood capillaries down concentration gradients. So this is also correct. 3. The relatively low concentration of carbon dioxide in the alveolus results in carbon dioxide leaving the red blood cell, also against concentration gradient. To combine with allowing hemoglobin to combine with oxygen, this is correct. The answer is going to be A. 36. Goblet cells are found in the trachea. Which cell structures would be found extensively in a goblet cell? So, as we know, goblet cells release high proportion of mucus. Therefore, it's very normal to have Golgi body, because mucus is basically a glycoprotein, and Golgi body modifies protein by adding a glucose unit to it. So this is correct. Here we have mitochondria. Obviously, to be able to synthesize that much protein to make that much mucus, it does require a high proportion of mitochondria, so this is correct. In order to synthesize proteins, high proportion of ribosomes are also required, because ribosomes is where mRNA binds, and that's the site of translation of the mRNA. So A is definitely going to be correct. The answer is 37. In a country where malaria has successfully been eliminated, an outbreak of malaria can occur years later. What could allow this later outbreak of malaria to occur? 1. Mosquitoes become resistant to insecticides. First of all, for those who don't know, mosquitoes is the insect vector carrying Plasmodium, which causes malaria. So it's an insect vector. So it's very normal if mosquito numbers increase and the insect vector increases, there's going to be a lot more malaria. Therefore, 1 is definitely correct. For 2, migration of the population due to war. This is correct because the more people there, the more likely there's going to be a spread of malaria from infected to uninfected people, so this is correct. 3, malarial parasites become resistant to quinine. Quinine is basically the anti-parasital medication used to treat malaria. So obviously if malaria became resistant to it, the amount of malaria increases, so this is correct. So all these factors allow an outbreak of malaria to occur, so the answer is going to be A. 38. Which row about a person with leukemia only and about a person with measles only is correct? White blood cell count in a person with leukemia only. So basically leukemia is the uncontrolled division of stem cells producing a higher amount of dysfunctional lymphocytes or white blood cells in general. So because there's an uncontrolled division, there's definitely going to be an increase. Here, definitely the bone marrow function will increase because there's an increased or aka uncontrolled division. 3. White blood cell count in a person with measles only. So a person with measles, that's a pathogen, enters the body. Macrophages carry out antigen presentation, and then B lymphocytes with receptors complementary to that pathogen divide. First of all, there's clonal selection, then expansion, to produce a higher number of those B cells that are complementary to the measles. Therefore, it's normal when an infection happens with measles that the number of white blood cells increases, so this is correct. The answer is going to be D. 39. What is the cause of symptoms of people with myasthenia gravis? Basically, this is an autoimmune disease where antibodies are produced against the neuromuscular junction and not allowing neurotransmitters to pass through, causing the impaired muscle movement. Okay, so the only correct answer for this is definitely going to be A. Antibodies block receptor molecules on the cell surface membrane of muscle cells, enabling its movement. However, for the syllabus for 2022, myasthenia gravis has been removed from the syllabus, so you don't need to know this at all. We just said it just in case. 40. Tetanus is a bacterial infection. The graph shows the blood antibody concentration of two people. On day 0, person G was injected with antibodies to the tetanus toxin and person H was injected with the vaccine for tetanus. What could be the result if G and H were infected with the tetanus bacteria on day 20? Okay, first of all, tetanus antibodies will not be produced with person G. This is incorrect because whether that person has passive immunity or active or artificial active immunity, it still would produce an immune response. This is definitely incorrect. By the way, this was a point of confusion for many candidates, so be careful to this point. B. Antibody production would peak after day 32 in person G. Definitely when clonal expansion, the primary immune response happens, antibody concentration definitely peaks when clonal expansion and clonal selection happens, so the answer is going to be B. Thank you very much for watching this video. If you like this video, please share and subscribe and support this channel. Thank you very much.